I wanted to talk about linkage disequilibrium, which is the correlation structure in SNPs. During my PhD, I analyzed a lot this because I was doing unsupervised genome-wide clustering. So I had to get a sense of what was the correlations I would find. So this is a package I wrote. It's mainly visualizations. There's 3 regions we're going to focus on. So this is the usual linkage disequilibrium plot you can find in genetic analysis studies. You will usually have on the upper side the minus log p-values associations. And on the lower side, you will have the correlation between SNPs. So for example, here each gray diamond is a SNP. And below in red is the correlation between 2 SNPs. And usually what happens is that when there is one SNP that is associated with a phenotype, you have several other around that are also slightly correlated with it. It might be less associated. And so this is also why we call it Manhattan plots, because it makes towers. This is chromosome 1. It's around PTPN22 gene. I selected it because I was studying autoimmune disease. This is the kind of correlations you find most generally. You can have like very well-defined triangles of correlations. So maybe 6, 7 SNPs that are very correlated. And we have other maybe 10 to 20 triangles, a bit less correlated, but we still have kind of the well-defined triangles like this. More or less well-defined. So this is the most usual correlations we will find. And this is a very interesting region. It's chromosome 8. It's BLK gene that is around here at the end that is relevant to autoimmune disease. So what's happening here is that it's an inversion polymorphism. So basically, in the population, you either have 101 or 010. Like it's a set of SNPs, but we all share either one sequence, either the other. So that's why we call it an inversion. It's either you have one or the other. And so we can see when we visualize the correlations that maybe 800 SNPs are very strongly correlated. So this is the kind of the exception. It's the most large inversion. There are a few other inversions.
What people do with this kind of situation? They trip. What I meant is how do they trip? Because in GWAS kind of thing, they some do empty pruning.
So they do a haplotype modelization. And so basically all these 800 can be summarized by one variable, since it's a long sequence and most people have either one sequence or the other. You can just replace it by one variable, 0 or 1. So that's haplotype summarization. So yeah, we call these kinds of sequence of correlated SNPs haplotypes. And HLA is a good example for that. So we're going to HLA now. So HLA is probably the most important region. It's 3 genes that are part of the MHC region. MHC region is around 30 megabase pairs to 35 megabase pairs on chromosome 6. So here I zoom in on HLA is around here somewhere. So we know that HLA is very strongly related with geographical information, ancestry, ethnicity. And if you take the genome-wide data, so 100,000 SNPs or more, you do a PCA on that, you will basically get geographic information. And it's very precise. You can have French, European, Italian, you can even have departments between France. That's how the precision goes. And so these principal components are driven by HLA. So HLA is the most dense region in SNPs. So most SNPs per length. And we also have what we see here is that we have some strange correlation, because it's not like in the first case where we had well-defined triangles and not at all correlations between more far apart triangles. But it's not either like the second example, inversion, where everyone is correlated. Here it's more like, so we have some triangles, but we also have more broader correlations. I kind of call it large but diffuse correlations. It showcases that it's a challenging region to analyze. There are long-term correlation, meaning from SNPs far apart from each other, but it's not easy haplotypes like in inversion. This is a little example of how my package visualized the HLA region. We first have the RS SNPs, and I query Biomart to get the gene name. So here we have HLA DRA, DRB1, and DRQA1, DQB1.
The number of the number is the correlation between the two groups.
Yeah, it's the R2 value. So R correlation squared. And you can combine with association plots on the upper side. So for example, here we have this region that is kind of correlated. And so it maps up to here. And so it's this map. So usually.
So, Tom, quick question. The horizontal bar, where they are at is based on actual base pair, right? Because
Yes.
I see one SNP, for example, to the other, sometimes they're really far apart.
Yes.
Sometimes they're close because they are different, just physical. Right?
Yeah.
How many bases?
Is the physical. So I'm thinking that these are the kind of plots to troubleshoot, quality control your RA data. This is the kind of plots you would want to do. You put on the upper side your associations with RA, on the lower side your correlation between SNPs, and this will help you troubleshoot.
Because if you were to plot the entire HLA, right, you should know which gene, like how far apart, what's their correlation, and what association you see right top will be instead of p-value, you'll look at the cosine. In which case you might be able to tell what might have, because even just purely driven by correlation, if you observe anything in the literature, but that information in the data that we have, the only place that will show up is actually Nucleotide bank, because only in patient level data you'll see it, right? So, but I'm curious, Thomas, just a quick question about this visualization. How, I like it that you pair them up, but how is this visualization, the LD, because I've seen different visualization, because most of the genetic paper, they have to do this. So how is your like a package different from?
So it was especially to do large scale regions. And so the first figures were 500 SNPs. And in my PhD, there's larger ones also. And so it was especially to focus on large regions.
Large regions, I see.
And we have a few plots also for tag SNP quality control.
And in that case, the correlation R squared you are reporting, where did the information come from?
It's from the raw data, the SNPs.
Or the raw data, not like, for example, 1,000 genome or something.
Well, it's we had like 1,000 patients, and each patient had 300,000 SNPs. That's how we measure.
And the correlation, I think the highly correlated variants could most likely to have the similar phenotypic effects, right? Do you think so?
So usually what we see is that one tower in associations,
Yeah.
so like several SNPs that are close together,
Yeah.
will have a tower in association and a triangle on the linkage disequilibrium plot.
Yeah, I would almost use it as QC. When you have like a high LD block, then your p-value only a singleton standing up there, their neighbors, no signal. That's usually something from normal your data, right?
Yeah.
Just purely by correlation, you should expect them to have similar correlation. So we'll see like a very high p-value, like a few dots just in the same region.
There was actually a fun story. Google did a longevity study, so like which SNPs predicted your life expectancy, and they had a lot of batch errors. So it's when, I mean, there can be a lot of things going on wrong, but basically in the reported results, they had just individual SNPs and not towers, and it got retracted a few weeks after. Thank you for your time.
No problem. Thank you.